Good morning, students. So, today we are going to start a new chapter, that is Seeds, Structure and Germination. In biology, the term fruit has a much broader meaning. It is a fertilized ovary with seeds. Once fertilization takes place in a flower, the ovary ripens to form the fruit and the ovule forms the seeds. Broadly, the seeds are of two kinds, that is monocotyledonous and dicotyledonous. The seeds with two cotyledons are called dicotyledonous, and with a single cotyledon is called monocotyledonous. In some seeds, the developing embryo is surrounded by a nutritive tissue called the endosperm, which stores food and help the embryo grow. Seeds with endosperm are called albuminous or endospermic, whereas in seeds like mango, the endosperm is absent. Such seeds are called exalbuminous or non-endospermic. The outer covering is the seed coat and is made up of two layers, the outer testa and the inner tegmen. The seed coat also has a scar on its surface called the hilum, which helps attach the seed to the fruit. Above the hilum is the micropyle, a small pore on the outer coat of the seed. Through this micropyle, the pollen tube enters into an ovule, and even the seeds absorb water through this micropyle and make it available to the embryo for germination. Cotyledons mainly contain food for the embryo and protect it. If you separate the two cotyledons, you will see the tiny embryo is attached to one of the cotyledons. The embryo consists of two parts, that is, the radicle and the plumule. Radicle forms the root and plumule forms the shoot. Like dicotyledonous seeds, the outermost covering of monocotyledonous seeds is also known as the seed coat. For example, the maize seeds has membranous seed coat that is usually fused with the fruit wall. Monocotyledonous seeds can be endospermic or non-endospermic. The endosperm of maize is bulky and store food, while the embryo is tiny and placed in one end of the endosperm. The embryo is separated from endosperm by a proteinous layer known as aleurone layer, and the embryo consists of a single cotyledon hair called scutellum. It also has a plumule and a radicle. The radicle is towards the pointed end and it is enclosed in a protective sheath called coleorhiza. And the plumule is towards the upper border side of the embryonic region and is enclosed in a protective sheath called the coleoptile. Next is germination. Germination is the process of formation of a seedling developed from the embryo. For germination, water, suitable temperature, and oxygen are very much essential. After a lot of experiments, it has been found out that water, oxygen, and suitable temperature is essential for germination. If you keep some seeds on wet cotton wool for 1 or 2 days, you will find that the seeds have started germinating. But it will fail to germinate on dry cotton wool. It will show that water is necessary for germination. Next is, if you take some gram seeds on wet cotton wool and keep it in an ordinary room temperature, you will see after 1 or 2 days that the seeds have started germinating. But if you keep the seeds in refrigerator, it may not show sign of germination or may germinate after several days. This shows that a seed requires moderately warm temperature of about 25 to 35 degrees Celsius to germinate. Both very low and very high temperature are unsuitable for germination. Next experiment will show how oxygen is necessary for germination. If you keep some soaked gram seeds with alkaline pyrogallic acid, and you will find that the seeds are failed to germinate because pyrogallic acids can absorb oxygen. That's why seed do not germinate, because there is no oxygen. Next is types of germination. Germination are of two types, that is hypogeal germination and epigeal germination. Hypogeal germination means where the cotyledons remains underground. Here the epicotyle elongates. So epicotyle is the region of the axis between the point of attachment of cotyledons and the plumule. Whereas in epigeal germination, cotyledons pushed above the ground and the hypocotyle elongates. Hypocotyle is the region of the axis below the cotyledon. Here you will see the clear picture of hypogeal germination. Hypo means below and geo means earth. Take an example of pea seed. The seed absorbs water and swells considerably, due to which the testa softens and bursts. The radicle emerges out, grows downwards and forms the root system. And the plumule grows upward and forms the shoot of the seedling. Here the cotyledons remain underground. Next, take an example of bean seed, which shows epigeal germination. Epi means above and geo means earth. Here also the seed absorbs water and swells. The radicle grow downwards to form the root system. Here the hypocotyle grows and becomes straight, bringing the cotyledon above the soil. Therefore, the germination is epigeal. Now let's see how germination takes place in maize grain. Here also the grain imbibes water and swells. The radicle pierces through the protective root sheath called coleorhiza and grows downwards to form the root system. But it dies off soon. New roots actually develop from the base of the stem called adventitious roots. The shoot part grows straight upward through its protective sheath called coleoptile, and the scutellum absorbs food from the endosperm till it is exhausted. Here, the hypocotyle does not elongate, and the germination is hypogeal. Some plant shows viviparous germination, in which the seed germinates inside the fruit while it is still attached to the parent plant. And after germination, the plant drops the seedling into the soil, which develops root and fixes itself. So, what is seedling? A seedling is a very young plant that grows from a seed. Germination in any plant ends with the formation of a seedling.